Today, I would like to share some of the progress we've made towards the development of the optical cochlear implant. This is a combination of a gene therapy using viral vectors to make the neurons in the cochlea light sensitive, and then of a new medical device, which is the optical cochlear implant, that then stimulates those neurons with light instead of with current, like the current electrical implants would do. We hope that this will cause a transformation of hearing restoration with the cochlear implant. Let me just briefly introduce the ear, where you know about the outer ear, and of course the middle ear with the eardrum and the ossicles. And today we're going to focus entirely on the cochlea in the inner ear, where sound information is encoded into neural signals and then sent to the brain. We have to use animal experiments in order to develop the new optical cochlear implant that we will eventually translate to a human clinical application. To put the cochlear implant in place, I'd like to introduce some of the aspects of hearing. Precisely, how do we actually code frequency information in the cochlea? We have a place code. This is a map of the cochlea, where you see that the high frequencies are mapped to the very base of the cochlea, and then, the lower frequencies come. And at each of these positions, you have sensory hair cells in the normal cochlea that pick up the mechanical vibration and transmit the information at their synapses with the spiral ganglion neurons. If we record from single spiral ganglion neurons, it looks like that. What you see here are little discharges, potentials, action potentials, that are driven by the acoustic stimulus, which in this case is a low frequency tone that drives the spiking at very distinct places in time. The spikes in the neuron, that is the neural code, follow the acoustic stimulus with very high temporal precision. That's why we see these distinct peaks here in this histogram. This is a time code. I told you about the place code for frequency. The time code for low frequency sounds also contributes to our pitch perception or frequency perception. Now you see both things together, the time code and the place code, because you appreciate that each of these three exemplary fibers are perfectly tuned to just one frequency, where you need the least sound pressure to drive the firing. When I started my career working with hair cell synapses, I came across an unexpected finding. These synapses are very unconventional. They differ very largely from the molecular makeup of a brain synapse. And we think that this is necessary because the function of this hair cell synapse is just so amazing. It needs to operate at very high rates with utmost temporal precision over long periods of time. And so Mother Nature came up with a very specific sets of protein for these hair cell ribbon synapses, some of them being identified by geneticists such as Christine Petit in the case of Otto Ferlin. When you now think from the perspective of a hearing impaired, and if you think about how deafness arises, you could actually assign a specific form of hearing impairment to these cases where the synapse doesn't function. We call this a synaptopathy, because the synapse doesn't work properly. And I want to basically illustrate this concept by using otoferlin as an example. What you see up here is the domain structure of the protein, and on top of that, you see missense mutations that have been identified in the field. Christine Petit's group started the whole research on otoferlin by identifying mutations in deaf people and demonstrating, based by the structure of the protein, that most likely this protein is involved in calcium binding. Clinically, a synaptopathy would leave many of the upstream processes of the cochlea unaffected. We can see that in children, for example, the function of outer hair cells in such people with otoferlin mutations can be maintained. So only where synapses code the sound, the function is disrupted. But before that, like in outer hair cells, which amplify sounds, it is actually maintained. This is important because this indicates that we could perhaps replace the disrupted otoferlin gene via gene therapy and place it into hair cells that are embedded in an intact cochlea to restore hearing. What results, though, is that there is no transmission or very little transmission of signal across the synapse. And so, in a result, the compound action potential that you see here in a normal hearing human is gone in the patient with an otoferlin synaptopathy. How to work out the precise mechanism? I said it's a synaptopathy, but how do you figure that out? Well, you have to go and do an animal experiment. In this case, you do an animal model of the human deafness. In this case, otoferlin is missing. Otoferlin, in normal cases, distributes across the whole hair cell, but also is present at the hair cell synapse, where you see this dark structure, which is the electron-dense synaptic ribbon. And so on top of that sits the synaptic vesicles that probably carry otoferlin. In a collaboration then, Christine Petit's group and our group could show that if you disrupt otoferlin in these mice, the hair cells do not exocytose or output transmitter anymore. You see that here on the ordinate. This is exocytosis, this is time, and we stimulate these hair cells, which you see over here. And then in the wild type, you have a fast phase of exocytosis and a sustained phase, but in the mutant, almost nothing. And so this finding indicates that indeed otoferlin is required for exocytosis of synaptic vesicles so that the synaptic transmission towards this spiroganglion neuron can work. But we can even look closer, and by electron tomography, Caroline Wichmann demonstrated that otoferlin might be important for tethering synaptic vesicles to the plasma membrane. Precisely, short tethers, as seen here in green for the knockout, are heavily underrepresented in the case of otoferlin knockout. And so together, the research in the field has illustrated major and different roles for otoferlin, one probably being calcium sensing for fusion, one being preparatory steps before fusion, including this tethering of vesicles to the plasma membrane. And another one, which is important to couple exocytosis of a vesicle to its retrieval via endocytosis. So there is a multifaceted role for otoferlin. Now we understand otoferlin's function a little better. Still, much remains to be done in order to fully decipher it. Yet we are interested to figure out whether potentially we could use viruses to bring back the intact gene into these hair cells of the otoferlin patients. And indeed, this is possible. Groups in the US, in Paris, and also in Göttingen have managed to illustrate this successfully. What you see here is a cochlea of an otoferlin knockout mouse, and this one was injected with a virus, and actually 2 viruses that contain parts of the autofill encoding sequence. This is a so-called dual AAV approach, which was required because the coding sequence of autofill is so large that it doesn't quite fit into a single AAV. AAV is adeno-associated virus. So what you see here is basically rescue of expression of autofurlin in these hair cells. And not just the protein was present, but also the function was partially restored. So what you see are auditory brainstem responses in normal mice, and over here you see that there are no such potentials in those autofurlin mouse models that were generated to mimic the human disease. This little potential here comes from hair cells, not from the neurons. However, when Ellen Reisinger's group injected those dual AAVs with 2 different designs, they could restore these auditory brainstem responses at least partially. And that's very promising for the sake of developing a gene therapy for autofurlin-related deafness for humans. To go a step further, we have aimed to package the entire coding sequence in one virus to make the approach a little less complex. And indeed, what Vladan Rankovic and Christian Vogel could show is that there is also expression of autofurlin in these autofurlin mouse models when using this overloaded AAV. And as a matter of fact, auditory brainstem responses could be restored. So in blue, we see injected ear. In yellow, we see the non-injected ear, which had some potential because there was a spread of virus, reflecting difficulties in developing gene therapy towards the clinical translation. In this particular study, Nikola Strensky's group could also show that there is an auditory percept at the behavioral level in those rescued ears. So there is a response to basically an acoustic stimulus that she found in the rescued animals, but not in the non-rescued animals. This is a really interesting and exciting avenue for future cure of genetic hearing disorders. However, there is a huge number of people suffering from hearing impairment and deafness that has not resulted from a defect in a single gene. And the standard clinical care for those people is hearing aids and cochlear implants. The cochlear implant is a very impressive success story. It's a neuroprosthesis in almost 1 million people, and it allows those people to typically understand speech in the quiet, even via telephone. And the great thing about the cochlear implant is that it really helps no matter what the precise mechanism of the deafness is, as long as you have an auditory nerve that you can stimulate. The method is that you have a microphone, a processor that processes sound information and generates electrical pulses that are then transmitted via this coil to the implant and chipped along this electrode array right into the cochlea. And then the cochlear implant aims to take advantage of the place code that I was introducing earlier, where we directly stimulate the spiral ganglion neurons, the neurons in the cochlea, at the base for high frequencies, and so on and so forth, up to low frequencies in the apex. This is great, but it is still not good enough. And so there is a major clinical need for further improvement, which results from the wide spread of current from each electrode contact, so that instead of playing piano with fingers, you play piano with a fist. And in order to improve that, we reasoned, once optogenetics, the control of cells with light, came up, that we might replace the electrical stimulation with optical stimulation using optogenetics. And that is because light can be confined in space much more conveniently, we could arrive at a more fine-grained stimulation of the place code of the spiral ganglion neurons. However, it's challenging. It's a new medical device. It's requiring gene therapy. I want to basically give you a brief audio introduction for how cochlear implant hearing might sound. Please note that this is just a simulation. In one case of a 8-channel vocoded music piece, and in another case for optical implant where we have more channels available, a 128-channel implementation. We want to translate our progress made in animal experiments to the clinical application in an optical cochlear implant. I'd like to share now some of the progress. The first thing you gotta do is make those neurons light sensitive. And as I introduced, we use AAVs, adeno-associated viruses, to carry the light-gated ion channel that is sort of a light switch in the neurons to express it inside the cochlea. This is what Christian Frobel pioneered in the lab. It's a direct injection into the cochlea using a quartz capillary. And long and behold, we do transduce those neurons, and you can see the expression of the tagged ion channel in the membrane of the spiral ganglion neurons, not in the non-injected ear. Do these animals hear? Yes, they do. If we place an optical fiber into a natural window, the so-called round window of the cochlea, we can stimulate brainstem responses with light. As we increase the light intensity, those brainstem responses grow in amplitude and get faster. But is it better hearing than the electrical hearing with the current cochlear implant? And in order to ask how much better the spectral selectivity, that is, frequency information, via optical implant would be, Alex Dieter combined the optical stimulation at 3 places of the cochlea with recordings from the auditory midbrain where he inserted a multi-electrode array. The reason doing that is that this structure has frequency layers so that you can record activity at this spot if you have a low frequency acoustic stimulation, and that you record activity here if you have a high frequency stimulation of the cochlea. In other words, if now we turn on the light at the high frequency spot of the cochlea, we expect to have activity down here. If we turn on the low frequency light spot, then we should have activity here. And she can calibrate all that. This is basically the surgical cetus with 3 optical fibers, high frequency, mid frequency, low frequency. This is where the electrode has been. And then we use acoustic stimuli to calibrate the system. You see these tuning curves here for a low frequency, mid frequency, high frequency, and we can extract the tonotropy of the auditory midbrain from those calibration experiments. And here we now compare the tuning curves obtained with acoustic stimuli, which is natural hearing, and optical stimulation at these three different spots. And what I hope you can see is that these tuning curves are actually very similar to the acoustic normal hearing. Now, what happens if we implant a clinical style electrical implant? And that's what you see here. This is with monopolar stimulation, which is used in most patients to date. And you can appreciate that there is a little dimple here where for very low currents we get activation at the low frequency, mid frequency, high frequency. But then as soon as you increase the current level, you activate the entire cochlea. So there's very poor frequency selectivity, while with optical stimulation, we are closely approaching normal hearing. This gets a little bit better as we use bipolar stimulation with 2 electrodes being fired, which offers a little bit more spatial selectivity. But as you see, as soon as you go a little stronger, you're still activating the entire cochlea. But now we have great frequency selectivity, and we wonder whether the temporal code that I introduced initially is maintained or is impaired. And there could be reasons, like for example, that these light-gated ion channels close within milliseconds when you turn off the light. Can they provide the temporal precision we need for the optical coding? And because this was not the case for most available channels, we invested time in generating ultra-fast channel rhodopsins. And we embarked on two channels, one being Kronos and Crimson. And we made fast functional channel rhodopsins in the blue and in the red light range. And I just want to give you one little snippet of data for Kronos, which is the fastest occurring natural channel rhodopsin. And I want to give you the best case first. This is the fastest unit, where each dot again is one spike. And you can appreciate that for every stimulus, we get a spike. The same for 100 Hertz. And then at 300 Hertz, you can see that it actually fades out a little bit because the neurons can't keep up this pace of stimulation. But even up to 1 kilohertz, the neuron was from time to time responding with good temporal precision. Now, in the population, the average is more that we get reliable and very precise responses for 100 Hertz and also 200 Hertz. And then, the response kind of breaks down as you go higher in stimulation rate. Nonetheless, 200, 300 Hertz are about the near physiological firing rate. So Kronos enables those neurons to follow their near physiological firing pattern. However, Kronos is a blue light gated channel, and blue light is afflicted with potential phototoxicity, so light can be damaging. And that's why we invested in generating ultra-fast red-shifted ion channels. In collaboration with Ernst Bamberg, we targeted Crimson, which has an absorption peak at around 600 nanometers. And we generated fast and ultra-fast Crimson in order to have tools available for clinical translation with less risk of phototoxicity. Sure enough, we could express fast Crimson or F Crimson in the cochlea. We could also elicit auditory brainstem response with the light stimulus. And we found that at the single neuron level, F Crimson enabled near physiological firing. You see that here, 100 Hertz, every stimulus gets you a spike. At 300 Hertz, you have some adaptation, and then even more at higher rates. So now we have a promising red-shifted ion channel for optogenetics. We thought to go even a little step further. What about very fast Crimson? And this is work in progress, and I just want to share some preliminary data with you. This is using auditory brainstem responses, and we're comparing a trafficking optimized version and the original VF Crimson, and we can see that short light passes are sufficient to trigger response. We can also go at very high rates, up to 500 Hertz, and still have these auditory brainstem responses. So it really helps to use very fast channel rhodopsins to drive the auditory system. And what about at the single neuron level? Well, up to 100, 200 Hertz, things are very well behaved. We have good temporal precision of firing. Thereafter, it's degraded. And the same happens for spike probability. So you get less spikes per pulse if you go for very high rates. All right. So let me then spend the last minutes talking about technology development. And what I want to share with you are two approaches that we take in parallel to work out LED-based optical cochlear implants and waveguide-based optical cochlear implants. And I will explain why we do this. We started with gallium nitride micro-LEDs because this is a very established technology. And those were embedded on wafers onto polyamide carrier with lines towards these LEDs. You see here one of these LED implants on the wafer with many of these LEDs, 144. And these optical cochlear implants are so flexible in one dimension so that we could implant them even into very tiny cochlea of rodents. This is a very strong technology, but it's mostly available in the blue wavelengths. In order to target redshifted opsins, we decided to also develop waveguide arrays together with our collaborators in Chemnitz, which you see here at the electron micrographic level. These are individual waveguides, and these are structures that couple out the light towards the neurons. And we then connect them to laser diodes, which are not plotted here, that feed the light into the waveguide. And then the waveguide pipes the light into the cochlea. And the advantage is that we have a very robust technology inside the cochlea, which is just polymer waveguides. Whereas it's much more complicated to safely encapsulate micro-LED-based implants inside the aggressive salt solution of the cochlea. No matter which of these passes you use, you will need the hardware to drive the emitters. And that's what we developed here in Göttingen, where you need to capture the sound, you need to process the sound, you need to drive the LEDs or laser diodes. And this is something that I want to briefly describe to you by saying that we need very low weight because we're working with small animals. We need this to be stable because, of course, if it's a medical device, it's got to be stable and needs to be functioning over many years. And of course, it needs to be power efficient so that you don't need to change your batteries every now and then. We want, of course, to be fast and wirelessly controlled and use a broad bandwidth in the frequency range. And then, eventually, we want to compare our new medical device, the optical cochlear implant, to the current electrical cochlear implant to really figure out how much more efficient it really is in conveying auditory information to the brain. And that's why we developed this hardware for both the optical and the electrical implant. Let me just briefly tell you what you see here. These are printed circuit boards that have all the driver electronics for the optical cochlear implant and the electrical cochlear implant. And up here, you see the same digital signal controller for both sorts of implants. And then they use an antenna to pick up the wireless control, and you have a battery on top. And then this whole assembly is encased in this plastic case, very light. And it's connected to the implant itself, which is either an optical implant or an electrical implant. Now, what did we do with that? We used rats to implant the system. In the first case, I show you data for an LED-based cochlear implant using very powerful and large LEDs. And so here you can appreciate that each of these LEDs is roughly 250 microns. It's encapsulated in silicone, and it's pretty robust. You can control every LED individually. You can bend the whole implant as you would in the cochlea, and it still functions. This is how it looks when it comes to us. It gets implanted, and then basically we use a round window to insert the optical cochlear implant, just like it's done in the clinic for electrical implants. And we can show you that it's inside the cochlea based on X-ray tomography, where you see these LED pads inside the cochlea. And next, I want to just briefly share with you the first results we got with this, where we again use the auditory midbrain to read out the frequency selectivity. That's shown here. This is the tuning curve for an individual LED, and that's the tuning curve for an individual electrode with monopolar and with bipolar stimulation. And so just as I indicated before, using optical fibers, now we use LEDs on multi-channel optical cochlear implants to drive the neurons of the cochlea with good spatial precision. That means with good frequency selectivity. And you can see the difference between electrical coding and optical coding down here. Can we do that at the behavioral level? Can we restore hearing in deaf animals? And yes, we can. So first thing to do is to bring the virus to the ear so that the light switch gets into the neurons. Then we would train the animals acoustically and also record the brainstem responses. Then we would basically deafen the animals to generate a deafness model using aminoglycoside treatment. And then basically we would insert the optical cochlear implant. And as you see here, this is the acoustic response before deafening, and after deafening there is no acoustic response, but there is an optical auditory brainstem response. At the physiological level, animals hear. What about behavior? So we have pre-trained the animals using acoustic sounds before deafening, and after deafening, the animals could right away perform the behavior with the optical stimulation, as if it was similar, as if it could be generalized. We went a step further and used the whole system and a sound coding strategy that is currently used in electrical cochlear implants to then use sounds to drive the same behavior. And that's what you see here. So these animals have been deafened, and then upon the sound stimulation, they could perform the behavior. So yes, the animals can hear the light. There is restoration of hearing using optogenetic cochlear implants. As a final set of data, I want to share something really exciting and new, which is using these micro LEDs, which are just 50 micrometer in size, and so can be scaled up in number up to more than 100 individual micro LEDs. And so now you're experts in auditory midbrain tuning curves, and you can appreciate that in this case, a block of 4 micro LEDs inserted into the apical part of the cochlea elicited a nice tuning curve. If we did the same thing with mid-cochlea, same thing, and so on and so forth. So now we needed 4 micro-LEDs because a single micro-LED would not reliably trigger a response. So we need to work on better emission of these probes, and I want to share this with you, how we're going about that. But before I do, I want to basically remind you of the difference between electrical stimulation and optical stimulation. This is what the micro-LED implant looks like in the cochlea. Now, how can we get more light to the neurons from the same micro-LEDs? One way is to put optics on top of these LEDs. And that's done by the group of Patrick Ruther, our long-term collaborator. And in this slide here, you can see one LED without such optical device and one LED with this optical device. And I think you can appreciate the height and the narrow beam profile of the LED aided with this optics. And that's just summarizing that in quantitative terms. With this, I'd like to conclude. And I would like to state that we demonstrated in rodents that optogenetic stimulation of the auditory nerve is feasible. I think I could convince you that we reach better frequency selectivity using optical stimulation than we can achieve with electrical stimulation. In fact, it gets actually pretty close to the natural hearing. We have worked towards ultra-fast channel absence in the blue light and in the red light. And so we think that better spectral selectivity can be accompanied by good temporal precision of optical coding when using these ultra-fast channel absence. And eventually, our collaborators and us have developed optical cochlear implant systems that we can now study and characterize in preclinical experiments before translating it into a clinical trial. With this, I'd like to acknowledge the people who actually did most of the work. we have been able to form a team out of biologists, medical doctors, physicists, and also engineers to actually make both the medical device and the gene therapy happen in preclinical experiments. With this, we could rely on our collaborators in different places, in Frankfurt for the opsins, in Freiburg and Chemnitz for the medical device, and we also collaborated with MedL company who provided electrical implants for the benchmarking of our optical cochlear implant. I'd like to stress that this work would have been impossible without the generous support of the European Research Council through the grant Opto here, and the German Research Foundation, primarily through the Leibniz program, and the federal government. I'd like to say that we are ready for commercializing the technology in the newly founded Optogentech company. And before I close, I want to share with you 2 videos which kind of illustrate progress in the last years. So the person you're seeing playing the piano is my son. And it's been in 2017 when we had the first version of our demonstrator with a micro LED chain and a transparent cochlear and our processor at that time. Just stop. And so now, in 2019, it looks like that. Same place.
Menschen, die nicht hören können, Musik in seiner ganzen Bandbreite zugänglich zu machen, ist die Forschung bisher nicht erfolgreich, anders als beim Verstehen von Sprache. Im Cochlear Inner Lab wird dazu ein neuer Ansatz vorangetrieben.
And our new model, which is a little bit more realistic. And with this, I'd like to close and thank you very much for your attention.